Talks. My name is Kate Fitzgerald. I'm a professor at UMass Chan Medical School in Worcester, Massachusetts, and I'm one of the organizing committees for a member of the organizing committee for this year's series of talks. So it's a real pleasure to welcome today's speaker. Before I do that, I just want to quickly mention that next week our speaker is going to be Ashley St. John from Duke University and also the National University of Singapore. So tune in next week. So today it's a real privilege to introduce a friend and colleague, Sarah Gaffen. So Sarah is currently the Gerald P. Rodnan Professor of Rheumatology at the University of Pittsburgh in the Division of Rheumatology and Clinical Immunology. Sarah received her bachelor's degree from Carnegie Mellon University, a PhD from the University of California at Berkeley, where she was working with Marion Koshland, a National Academy of Science member. She then completed her postdoctoral work also in California at UC San Francisco with the Gladstone Institute of Virology and Immunology. And then Sarah started her independent faculty position at the State University of New York in Buffalo in the Department of Oral Biology. And then has since that time, since 2008, then has been at the University of Pittsburgh in rheumatology in the Department of Medicine. So Sarah's research program is focused on the IL-17 superfamily of cytokine receptors. I think we're going to hear quite a bit about that today. She has focused in particular on the role of IL-17 in antifungal defenses, particularly in the oral mucosa, and also the role of IL-17 and the Th17 pathway in autoimmune diseases. And I think we'll hear about how targeting this pathway has sort of revolutionized our treatment of many autoimmune and autoinflammatory diseases. And then she'll talk about the consequences of these therapies on susceptibility to infection. So Sarah has received numerous awards. She's a recipient of an NIH Merit Award. She was a recent recipient of the BioLegend William E. Paul Award for Excellence in Cytokine Research. And she's also the president-elect of the International Cytokine and Interferon Society. She's an elected fellow of the American Academy of Microbiology and also the American Association for the Advancement of Science. So a lot of accolades. So we're really looking forward to Sarah's talk, but just before we get started, so as part of these global immunotalks, we usually ask the speaker sort of a question about their own personal career or their lessons they've learned along the way. And I wanted to ask you, Sarah, could you tell us a little bit about sort of a particularly impactful decision in your career and how that sort of led you to where you are today?
Thanks, Kate, and thank you for that very kind introduction. I knew this question was coming, and so I put a lot of thought into it. I think you alluded to my first position being in the Department of Oral Biology at the University of Buffalo, and that was in the dental school. And I think, looking back on it, the most impactful decision that I made in my career was to accept that position rather than a couple of other opportunities in more traditional microbiology, immunology departments. And that really steered me in the direction of looking at oral mucosal immunology and some of the, I would say, kind of neglected diseases, at least from an immunology perspective. And so my lesson to the trainees from that is that you can do interesting science it doesn't have to be the traditional immunology or whatever your field is. And I think there are opportunities out there in less studied areas. And so I mean, you see a lot on academic Twitter about how there's no jobs, and I don't think that's as true as people think. So broaden your horizons and take inspiration from the colleagues around you.
And now that you are where you are, at the time, was that a strategic decision, or do you think in hindsight it influenced, the trajectory of your own career?
Oh, definitely. It's a lot of this is 2020 hindsight.
Yeah.
But I will say, when I was choosing among positions, it was hard to put my finger on why it felt right to go to Buffalo. but I never regretted that decision. And I think I always tell people, follow your gut or your heart, or go, whichever organ you attribute those feelings to. If a place is supportive and feels good to you, then you will do well.
Yep, the gut test is central in my view. Cool. Okay. All right. Let's so Sarah, why don't you share your screen and let's make sure your slides work, and the floor is yours. Thank you again for sharing this work.
Great. Thank you, Kate. Does that look good?
That looks perfect.
All right. Let's get our laser pointer going. All right, here we go. All right. Well, again, thank you to the organizers for the invitation. It's wonderful to be here. I'm going to be talking about IL-17, of course, but being a global immunotalk, I thought I would open with a little bit of geography. Pittsburgh is here. We're in Western Pennsylvania. It's not necessarily the biggest tourist destination in the US. This is just for context, this is Philadelphia here, about a 6-hour drive away. Washington, D.C., which is actually where I currently am talking to you from, about 4.5 hours. Here's New York City, and here's Buffalo up here, where I started my career. So what is Pittsburgh known for? Many, many things, but here are 3. Of course, we have a football team, and no one tells you this until you move to Pittsburgh, but when you move here, you have to sign a contract in blood that says you'll become a football Steelers fan. So, there you are. Heinz Ketchup, we are the home of Heinz Ketchup, and also the home place of the pop artist Andy Warhol. And so if you're paying attention, there is a reference to Andy Warhol in my talk, and see if you catch it. This, of course, is my favorite intersection in Pittsburgh, the corner of Sarah and 17th Street. And I think you can see in the right-hand corner, even our Steelers logo. So that's just proof that it was, in fact, taken in Pittsburgh. All right, with that, as Kate said, I've been working on IL-17 for a number of years now. IL-17's been around longer than many people realize. It was cloned in 93 and its receptor in 95. And I started working on it in my own lab in 99. And literally at the time, all the papers on IL-17 fit in a single folder on my bench. Obviously, all of that changed in 2005 with the really seminal discovery of a new subset of T helper cells that made IL-17, eventually becoming recognized as a genuine lineage, so-called Th17. And of course, the interest in these cells, and consequently the cytokine, really exploded. And so that's sort of how we got our start. Now, what's different about IL-17? Well, as I alluded to, its receptor is really a distinct family of receptors compared to other cytokine receptor families. So it's not a TNF receptor, it's not a type 1 cytokine receptor. And the same is true of the ligands. These really constitute structurally and to some extent functionally distinct cytokines. The IL-17 family specifically has multiple receptors and ligands. And when I say IL-17 in this talk, I'm really referring predominantly to IL-17A, which was the founding member. It is close cousins to a separate cytokine encoded on a separate gene called IL-17F. These form homodimers and heterodimers, and they all predominantly signal through a dimeric receptor composed of IL-17 receptor A partnered with IL-17 receptor C. But you can see on this diagram that there are other configurations of the receptor that have been described for these ligands, perhaps some specificity there. And there are a lot of other ligands. Despite their names, these are encoded on separate genes. You can see there's a lot of question marks, even in just the simple question about what are the ligand receptor relationships. We have an orphan ligand in IL-17D. There are IL-17B receptors really not defined. So there's still a lot to do. And I encourage anybody interested in thinking about what they're going to do in the future to realize there's a lot of holes in this family as well as many others. So some of the very early insights into how this receptor family might operate came from, I think, a very neglected paper that I'm indicated here, Nováčková et al. And they were, it's really kind of a theoretical paper that suggested that the IL-17 receptor might be or was distantly related to the toll and IL-1 receptors. And this was predominantly observed in the cytoplasmic domains of these receptors. So there was a homologous motif found in IL-17RD, which at the time was known as CEF, IL-17RA, as well as some of the other receptor family members. And this motif had distant structural relationships to the so-called tear domain found in toll-like receptors and IL-1 receptors. Now, many of you will know that the MIDI88 adapter is the key adapter of the TLR and IL-1 receptor families, and it also has a tear domain. And this original paper suggested that the rather poorly understood adapter called ACT1 also had a CEFR domain, and suggested that perhaps ACT1 might be an important member, signaling molecule in this family. And that indeed turned out to be the case. So work from Chen Dong and Xiaoxia Li demonstrated that ACT1 was essential for mediating signals probably through the entire receptor family, although again, that has not been directly proven. And that it has a lot of very interesting functions. In addition to being an adapter, Xiaoxia Li has shown that it's a ubiquitin ligase, an RNA binding protein. And so it's a really fascinating molecule that is essential for at least for all of IL-17A signals as far as we know. In my talk, you will see allusions to ACT1 and ACT1 deficient mice, but really the majority of the work in this space has been done by Xiaoxia Li. So I encourage you to look at her work. My lab is really centered, we think of IL-17 receptor as being the center of our world. Sometimes we look at the cells that make it, and you've heard a lot about that in some of the prior global immunotalks in this series. But predominantly our interest is in the cells that respond to IL-17 and how they do that and what are the consequences of that. And with that, most of the cells that are IL-17 responsive are non-hematopoietic. They're epithelial, fibroblast, other types of mesenchymal cells. And the consequences of signaling in response to IL-17, of course, it evolved to protect us from infection. And a lot of my work has been to look at the role in immunity to fungi, and particularly to oral fungi, namely candidiasis. And that'll be the first part of my talk, but I'm only going to talk about that briefly because there's actually another webinar series that I've done which I'll show you a link to that talks more about this. The majority of my talk today is going to be on the flip side of IL-17 signaling, which is how it drives autoimmune pathology. It was clear very early on that IL-17 was popping up in autoimmune conditions. And on the strength of that, a number of companies in the pharmaceutical world were developing reagents to target both IL-17 and its receptor. And these showed marked success in psoriasis. I'll talk about that a little bit. And of course, side effects do come along with that. But understanding how the receptor signals and mediates all of these different events and whether they're really all the same or could some of them potentially be exploited perhaps for therapeutic benefit, these are all things that our lab is interested in. But before I go further, I'm going to tell you a story, a case, as it were. This is a story about Rachel Bailey, who was an undergraduate in my lab during the summer a few years ago, and she later came back for a year as a tech. It's really a story, actually, about Rachel's mom. And so her mother had psoriasis, psoriatic arthritis, really most of her adult life. And it was never well controlled, unfortunately, until the advent of the anti-IL-17 biologics. And for her, as for many, this therapy really was a game changer. Her plaques cleared, she felt better. And I would love to be able to stop the story there. Unfortunately, she developed a number of adverse side effects, including pretty severe and recurrent oral candidiasis. This was initially thought to be a pretty rare side effect with this therapy, but it's a lot of data is emerging showing it's much more frequent than had originally been estimated. But she also developed a fairly severe inflammatory bowel disease, and this has made managing her condition rather difficult. I think this does illustrate in a single individual all of the many aspects of IL-17 function and, to some extent how little we know, but also how far we've come in the last 20 or so years. So as I said, I'm going to start by talking a little bit about the work we've done on candidiasis and particularly oral candidiasis. So what is Candida? This is a commensal fungus. It's a dimorphic fungus that can exist in yeast and hyphal forms. And it's this invasive hyphal form that really triggers disease. The most common manifestation of disease is oral candidiasis. This is a very common side effect in HIV/AIDS, anti-T cell therapy, but also inhaled steroids, infants, the elderly. So it's a common disease of immunocompromise. It can, in more severe manifestations, also cause dermal, fingernail disease, very difficult to treat, vaginal candidiasis, which is also very common in women. And when it becomes invasive, which can happen if one gets a bloodstream infection, this can cause organ invasive disease, which is accompanied by as much as a 50 to or even 80% mortality rate. Very challenging to treat. Fortunately, fairly rare and not associated with or at least the risk is not associated with mucosal disease, but nonetheless, this is a pretty severe problem. And I just want to point out, there are no vaccines to any fungal pathogens whatsoever. And so we really, I think that illustrates how little or how far we have to go in understanding the immune responses. The first hint that IL-17 might be involved in immunity to Candida was actually quite early, before Th17 cells were described, which was a suggestion by Fidel and Schwarzenberger that IL-17A or IL-17 receptor A actually might be important in systemic candidiasis. And this has been borne out by many in the field. And really, that was sort of where it was. But being in a dental school, I thought, well, it would be interesting to start looking at this in manifestations, oral manifestations of Candida. And so we did. And when I say we, I really mean Heather Conti, who's a faculty member at University of Toledo now. And what Heather did was, in our first foray into Candida work, was to show that mice lacking the IL-17 receptor, also IL-23, but not the IL-12 receptor, were very susceptible to oral candidiasis, suggesting that it was the IL-17 or Th17 axis, and perhaps not the Th1 axis, as had been suggested, that was critical for immunity to mucosal host defense. And we followed this up in many studies. This was actually this is what it looks like in a mouse. You can see fungal plaques on the tongue of this mouse on a journal cover that we got in Infection and Immunity a few years later. And we later showed that IL-17 receptor deficient mice, IL-17 RC deficient mice were also susceptible, ACT1 deficient mice were susceptible. And so this really gave us a nice system to be able to interrogate both IL-17 receptor signaling in the context of fungal immunity, but also to understand various tenets of oral mucosal principles. And what was really great was a couple of years later, Jean-Laurent Casanova and Anne Puel identified cohorts of humans who had similar mutations. So they had families with unexplained chronic candidiasis who turned out, many of them were missing some of these same components. So they had IL-17 receptor A deficient humans, RC deficient humans, ACT1 deficient people. And that really told us that we were kind of on the right track. Over the years, we've done a lot to try and understand what IL-17 is doing. And this is, I'm just going to summarize here because, as I said, I want to focus a lot of this talk on autoimmunity work. But one of our early approaches to this was to do just gene profiling of oral tissue. So looking at tongue in wild-type mice that have been infected with Candida compared to mice that are susceptible, namely IL-17 receptor deficient mice, we got a list of genes, as one does. Top on that list was beta defensin 3. This is known to have anti-Candida functions, and also a lot of genes that regulated the neutrophil axis, CXC chemokines, GCSF, et cetera. And we know that, so putting together work from us and many others in the field, we've shown that Candida, when it becomes hyphal and begins to invade the epithelial tissue, it triggers a lot of sensing in this epithelial tissue that activates a type 17 response. Some of these signals lead to activation of an adaptive response over time, but mice, unlike humans, don't carry Candida as a commensal, and so we also were able to visualize the early innate response. And so various signals, including a pore-forming toxin called candidialysin, trigger activation of innate IL-17 producing cells as well. And these are dominantly gamma delta T cells and even more frequent alpha beta T cell receptor expressing cells that have innate activity. Sometimes it's called natural TA17 cells. And cumulatively, the IL-17 made by all of these cell types acts on the IL-17 receptor on the epithelial tissue and drives these types of host defensive responses composed of beta defensins, which we've shown to be absolutely non-redundant, as well as the neutrophil response and monocyte response through CXC chemokine recruitment. So all of that that was a sort of a very quick overview. It's reviewed in this review article that with Nikki Mitsopoulos' group. This was our attempt to get a journal cover. This was a failed journal cover attempt, but I'm very proud of the picture. This was drawn by Rachel Bailey, so I wanted to give it a little bit of airtime in this talk. And if you're paying attention, that's the Andy Warhol reference. And finally, if you really want to hear more and you're very excited about fungi, I will direct you to another webinar series that I helped create with some colleagues at the University of Exeter and others called Mycotox. You can hear me, but you can also hear a lot of others in the field talking about their perspectives of immunity, not only to Candida, but to other fungi as well. Okay, so with that, I want to move on and start talking about some of our work on IL-17 signaling and how that pertains to autoimmunity. I already mentioned psoriasis, IL-17 blockade. There are a number of drugs that block IL-17. This is a little out of date, actually. There are still more out there. And these biologics are great for some diseases, but they've also not been so great in others. And understanding which diseases to apply these to, I think, is still really very much an open question. Now, biologics are great in some ways, but they're also very expensive and they're not accessible to everyone. And also they block everything that the ligand does. And so a lot of our premise is that by defining the downstream molecular signaling that this receptor activates, we might be able to get perhaps more accessible drugs, drugs that could be given orally, perhaps cheaper, but also perhaps more selective, with the ideal, of course, being that we could block signals that would provoke autoimmunity while sparing those that allow for host defense. And of course, the premise of defining cytokine signaling pathways and leading to drugs is not just pie in the sky. When I started my career, this was the beginning of the JAK-STAT era. And of course, JAK inhibitors have become used in very widespread manner for not only autoimmunity, but cancer and so forth. And the same thing for NF-kappa B inhibition, et cetera. So mostly when you start talking about signaling and molecular signaling, a lot of people's eyes begin to glaze over. This is sort of the principle that I always view with signaling is you have a complicated formula, a miracle occurs, and then you get an output. And of course, the goal is to try and be a little more explicit in what's going on in the middle. And when we started working on IL-17 back in 1999, this is sort of where we were. You could buy IL-17 from companies because it had been cloned for a while at that point. There was one known receptor subunit identified, that was IL-17RA, and one or two genes had been defined. And, otherwise there was sort of this black box in the middle where some sort of miraculous thing was happening and we had no idea what it was. And I presented this to my first student, Matt Reddy, and I said, let's go to it. And he said, great. And so Matt's reasoning was, if we're going to try and work out, a circuit like this, we need to have endpoints. We need lots of endpoints so that we can start to work backwards and ultimately, maybe meet in the middle from the proximal activation to the distal. And he talked me into doing what at the time, in 99 or 2000 was a very expensive experiment, which was to use the Affymetrix gene chip to get a really broad, unbiased view of genes that were induced by IL-17. And just parenthetically, we were working with fibroblasts because we had gone through every cell in the tank and none of the hematopoietic cells were responding. But at the time, we were working with an osteoblast cell line that did respond. And we said, great, okay, we'll stick with that for now. And so I'm not going to go through all of the gory details of that, but there were, of course, we got a long gene list and we were trying to work through what that meant biologically. But there were 4 genes on that list that have become the through line of our work ever since. The first is IL-6, and that had already been known. So that was a nice confirmation that this system was working. I don't think I need to introduce it to this audience. One of the most potently induced genes that we saw in these fibroblasts was lipocalin-2, also known as NGAL or 24p3. And it has both antimicrobial properties, particularly against gram-negative bacteria, but it was also a biomarker of kidney damage. And that was something that was in my mind. And you'll see as later in the talk that we have been working on autoimmune glomerulonephritis conditions, in part inspired by the discovery of lipocalin. Another gene on the list was CEBP delta, cat enhancer binding protein delta. This is a transcription factor that has been around for a long time, but certainly compared to NF-kappa B or AP1, I think is perhaps a little bit understudied, or at least poorly understood. And finally, there was a non-canonical member of the NF-kappa B family called I-kappa B zeta, originally called male. And we didn't work too much on this in the beginning, but this really began to emerge by many in the field as a downstream activator of IL-17. And you will see in my basically the rest of this talk, how these transcription factors and these target genes interweave to illuminate how IL-17 signaling really happens. Okay, so that very first paper, in retrospect, sort of looks a little bit simple, but basically what we showed was that IL-17 not only activates CEBPs, that included both CBP delta, but also CBP beta, and these work in concert in many respects. It induces these, and that these are required for induction of target genes like IL-6, and this is an old-fashioned northern blot for the kids in the audience. And if we look at the IL-6 promoter, which was kindly given to us by Oliver Eichelberg, this promoter is indeed activated both by IL-17 by itself, but also cooperatively with TNF. And I won't talk about signaling synergy today, but that is an important feature of IL-17 function. Happy to discuss it on Twitter. Activation of the promoter did not require the AP1 site, but it did require both CEBP and NF-kappa B because mutations of those sites in this promoter failed to be activated. And so we also looked at some of the other genes on the list, for example, lipocalin-2, and we found very parallel findings. This is work done by Feng Shen together with Matt Reddy. And we did, again, we cloned up the lipocalin-2 promoter, and it has various conserved sites. But the key sites were both NF-kappa B and CEBP. And so if we either deleted down to those sites, that's what's shown here, all of these retain those sites, these mutations do not, or if we individually mutate them, we lost activity. And so again, it seemed that the key modules that respond to IL-17 are both the NF-kappa B, what turns out to be I-kappa B zeta responsive elements, as well as the CEBP elements. And both of these are essential. That's, I would say, is how defining this early IL-17 gene signature really taught us something about signaling, and that the CEBP, here's CBP beta, delta, and I-kappa B zeta that get activated by IL-17. And we know that this, now that this is dependent on both ACT1, which activates TRAF6. And I think this sort of pathway is what a lot of people think about when you say signaling. You turn on adapters, you lead to transcription factors, and boom, those transcription factors work their magic, you get RNA and you're done. Now, a lot of genes on this list encoded other elements. And in particular, there were a lot of RNA binding proteins on this list. And I think it's important to remember that a lot of activation of gene expression is not just transcriptional, but is also post-transcriptional. So the RNAs that result from mobilization of these transcription factors is itself subject to a lot of post-transcriptional control. And RNA binding proteins can bind to these RNAs, both typically in the 3' untranslated region, but also in other areas, and control the fate of that RNA. That could be stability, that could be translation. It can even be earlier in terms of nuclear export or splicing. So there's, and I'm not even talking about mirrors. And so there's a lot of ways that RNA can be controlled completely independently of transcription. But there's also crosstalk between these pathways because, as I'm going to show you, a lot of these genes that we were seeing that are controlled by RNA binding proteins turn out to encode transcription factors. So the first vignette I'm going to tell you is about an RNA binding protein called regnase-1 that regulates I-kappa B zeta. All right, so what's regnase? This really came up because the gene ZC3H12A was on this list. So if we go back, here's ZC3H12A. And when you get a gene name like ZC3H12A on an array, I don't know about you, but I just sort of roll my eyes and ignore it. But fortunately, my student, Abhishek Garg, came to me one day and he said, Sarah, did you realize that ZC3H12A encodes MCP-IP1? And I said, I'm your boss. I know everything. Of course I knew that. Why don't you tell me what you know about it? And so for the trainees in the audience, you know, when your boss says that, that's now you know what they're really saying. So ZC3H12A had been identified in 2006, as the name suggests, as an MCP1 induced gene, and it was later renamed as regnase. But in its early days, it had been demonstrated to be a negative feedback inhibitor of TLR4 signaling, and given the relationships of TLR signaling, or TLR structure, at least to 17 receptor, we always sort of keep an eye on that. It had been suggested to deubiquitinate TRAFs, but I think its dominant function is as an endoribonuclease, and it induces decay of RNAs that it binds to, including IL-6. And it does that by binding through the 3' untranslated region. And so what Abhishek showed was that IL-17 induced expression of regnase very directly and very rapidly. And you can see that here. This is in ST2 cells, which is a fibroblast stromal cell line, that ZC3H12A, encoding what we now call regnase, was induced very rapidly. This was true at the protein level too. And if we have a deficiency of regnase, that signals are markedly enhanced. So you can see much stronger activation, for example, of IL-6 than you do in cells where regnase is intact. And so therefore, regnase seemed to be a potent feedback inhibitory signal in the IL-17 pathway. And so just to summarize a lot of the work that Abhishek did, he showed that a lot of the genes in the IL-17 pathway, so that regnase binds to these RNAs and degrades them. And this includes RNAs that directly drive inflammation, like IL-6, but also various chemokines. Interestingly, regnase also regulated the IL-17 receptor RNAs in certain circumstances. And again, I'm not going to go into that, but we can talk about it on Twitter if people are interested. And I think most significantly, what he showed was that regnase also degrades I-kappa B zeta RNA. Sorry for the nomenclature, but it's called NFKBIZ that encodes I-kappa B zeta, and that this was a 3' UTR dependent event. And so you can see that here, that if he co-transfected I-kappa B zeta that contains its 3' UTR together with regnase, we completely lost expression of I-kappa B zeta. But if we take away the 3' UTR, the RNA is expressed almost normally. And so consequently, everything that is I-kappa B zeta dependent, therefore, is affected. So, never give up, I think, is another theme in science. And so, we tried another journal cover. This is done by Nilesh Amatia, in a Pac-Man-like idea. And here's a regnase buzzing around, degrading things. Unfortunately, Immunity didn't like that either. Okay. So I've talked about some of the signaling. Now I'm going to move to, this was more molecular work, in vitro work, but does any of this function of regnase matter in vivo? And that brings me to a disease state that I think a lot of people don't tend to think about, which is autoimmune glomerulonephritis. And so AGN, as it's called, is a condition that is the terminal event of a lot of autoimmune conditions, some of which people debate if they're truly autoimmune, but it results from deposition of autoimmune complexes in the kidneys, particularly in the glomeruli. And a number of conditions can lead to this. They have very different upstream triggers, but the terminal events, and particularly the pathology within the kidney, are often very much shared. And the animal model for this, which was introduced to me by my close colleague Partha Biswas, so I'll talk about him in a minute, is demonstrated here. So in order to generate these complexes, we immunize mice with rabbit IgG and follow that with rabbit antibodies that target the kidney, and particularly the glomerular basement membrane protein, or GBM. 2 weeks later, we get pathology, and this pathology is characterized by a lot of standard things that we see in humans, including expression of lipocalin, lipocalin-2, and IL-6, but also all of these various parameters that are seen in the most severe form of human glomerulonephritis called crescentic GN. And you can see that all of these are recapitulated in this mouse model. Now, my associate, Partha Biswas, who's my lab next door neighbor, is a real expert in kidney autoimmune disease, and he kind of introduced me to all these complexes, all these concepts. And work done by Partha, looking at the IL-17 receptor, but also the late Elise Siebenlust from NIH, who sadly passed away a year and a half ago, working with Act 1, demonstrated that the IL-17 axis is essential for the pathology in this condition. And you could see even deposition of antibodies, but no pathology in the absence of IL-17 signaling. More recently, it's been shown by a number of groups, but I think this paper is very worthy of a call out, that pathogen-induced tissue-resident memory cells, particularly IL-17 producing cells, are found in human AGN, and these amplify autoimmune pathology. And these are against various commensal pathogens, including Candida. This paper was more about staph, but also E. coli, all infections that can get to the kidney, and in some cases can amplify and help reestablish infection. So in looking at regnase, our hypothesis was that an insufficiency of regnase might exacerbate autoimmune pathology. And so most recently, we've been looking at this in AGN, and I'm just showing you one figure here, but this is the paper. And again, I really want to call, this was a very close collaboration with Partha Biswas's lab. That mice that are lacking regnase, and this CRE is targeting renal epithelial cells, have much worse pathology than their littermate, wild-type littermate counterparts. This is measured by serum levels of blood urea nitrogen, which I'm showing you here, but every other parameter was also far more exacerbated. And this is not the only condition where regnase does this. In prior studies, we had looked at the mouse model of multiple sclerosis, namely EAE. I think you can see, it's actually blocked on my screen with the Zoom here, but I think you can see that regnase deficient or MCPIP1 deficient mice have much worse pathology in EAE. And the same seems to be true in psoriasis. This is actually human psoriasis, tissues we got from Johan Gudjonsson, elevated, very highly elevated expression, at least of regnase in human psoriatic plaques. And in the mouse model, we saw commensurately increased disease. And so not only IL-17A driven, but also IL-17C driven. So regnase seems to be a feedback inhibitor, not only in vitro, but also in mouse models of autoimmunity. But then one might say, but you're restraining signaling by IL-17. Maybe that's a good thing in immunity to infections where IL-17 is important. And indeed, that's the case. So we originally demonstrated that a number of years ago, looking at Candida albicans, that mice that are insufficient for regnase have a better response to systemic candidiasis because their IL-17 response is improved. And the same thing is true in Klebsiella pneumonia, recently shown in collaboration with Geraldina Trevejo Nunez, who has an independent lab now at Pitt in infectious disease department. And so one might say, and again, this is very speculative, but perhaps short-term blockade of an inhibitor of signaling like regnase could be a good thing in an acute infection setting, life-threatening infection setting, for example. So inhibiting the inhibitors might be a bad thing for autoimmunity, certainly, but could be a good thing in infection. Now, in all of these studies, there was a conundrum that kept popping up. IL-17 is inducing regnase as a negative regulator, and that induction seemed to persist even in the context of chronic inflammation. And I'm showing you one example here. This is done by Leticia Monin, who is a postdoc working on the psoriasis model. This is imiquimod psoriasis. It's not a perfect model of psoriasis, but it's standardly used. So mice in which we induce psoriasis, very strongly IL-17 dependent, show a strong increase in regnase expression. This is the RNA encoding regnase. And the same thing is true in humans. Again, Johan Gudjonsson gave us samples from psoriatic plaque. You can see regnase RNA, also protein is induced. But this is still in the face of inflammation that is IL-17 driven. So why is that? Is the inflammation still happening if this negative regulator is being expressed? So is something offsetting the negative effects of regnase? So that brings us back to this list of, in this case, Candida-induced genes, but these are IL-17-dependent RNA binding proteins called Arid5a. So this is my second vignette. Arid5a is an RNA binding protein that, as I'm going to show you or tell you about, actually has some capacity to counteract regnase and to promote expression of IL-17-induced genes. And the punchline is that it regulates not only IKB zeta, but also the CEBPs. So what is Arid5a? It was described originally as an AT-rich interactive domain protein. That's the origin of the name, and had been described as a transcription factor originally, binding DNA. Kishimoto's group in Japan demonstrated that it also binds to RNA through its characteristic arid nucleotide binding domain. And his lab had shown that it stabilizes, among other things, IL-6 mRNA by binding to the 3' UTR in a region that overlapped with the regnase site. And at least some of its function could be explained by counteracting the effects of regnase by competing at that site. Nilesh Amatia in my lab demonstrated that Arid5a was induced by IL-17, indeed, in many target cells. This is through NF-kappa B expression of Arid5a, and that Arid5a also positively regulated lipocalin-2, but it didn't bind to the lipocalin-2 RNA. And so it could activate the lipocalin promoter, as shown here. If he overexpresses Arid5a together with the lipocalin promoter, we see nice activation of that promoter. And this is dependent on CEBP and NF-kappa B binding sites in that promoter. So this data suggested that Arid5a might be regulating lipocalin, perhaps by regulating its upstream transcription factors. And so one of the experiments that Nilesh did was to see whether Arid5a actually binds to the RNAs encoding those transcription factors. And so similarly to what we had seen for regnase, he found that Arid5a bound both to IKB zeta RNA, but also CBP beta. And this is done by a procedure called RNA immunoprecipitation, or RIP, very analogous to chromatin immunoprecipitation, or CHIP, where one pulls down the binding protein of interest, in this case Arid5a, and looks, in this case, at the RNA that is co-associated. We did this with a MIC-tag version to facilitate the experiment. And what you can see is that with IL-17 stimulation, CBP binding to Arid5a is enhanced. And we, as I said, we saw the same thing for IKB zeta. And so presumably that would lead to expression of CBPs, which would then transactivate promoters like lipocalin. But we did have a bit of a surprise, because even though these bound to the RNAs of CBP beta and IKB zeta, they actually Arid5a didn't really change the level of RNA encoding these transcription factors. So here's this is the IKB zeta RNA. It's induced by IL-17, but loss of Arid5a really doesn't change that. And the same thing was true for CBP beta. However, if we looked at the protein levels, they were dramatically affected. So IKB zeta is very potently induced by IL-17 signaling and almost entirely abrogated, even with a partial knockdown of Arid5a. This is probably a 60–70% knockdown. So this suggested Arid5a could bind to the RNA, but it was not affecting its total amount, nor did it affect stability, which I'm not showing you. And what we found was that Arid5a binds to components of the translation initiation complex, specifically to the EIF4G scaffolding subunit, and that this facilitates translation, allowing translation of these transcription factors, which then, of course, in turn, influence expression of everything downstream. For other RNAs, we certainly showed that Arid5a could bind the 3' UTR and control their stability, for example, IL-6, which had already been shown by Kishimoto's group, as well as some of the other chemokines. So there's at least two functions by which Arid5a could mediate downstream target expression. We have subsequently made mice that lack Arid5a, and this is unpublished work, or well, I guess on bioRxiv, however you count that. And these mice are resistant to the IL-17 induced EAE model of disease. So this is autoimmune model of MS. Mice, littermates have a strong clinical score of EAE, whereas Arid5a knockout mice are entirely protected, similar to ACT1 deficient mice. And this had been shown by Kishimoto's group with an independently created knockout mouse. But what we were surprised by was that even though this very strongly IL-17 dependent autoimmune disease was behaving as we would expect, that candidiasis was not affected. So mice that lack Arid5a, as shown here, do not show any increased susceptibility to oral candidiasis compared to their counterparts, wild-type counterparts, whereas ACT1 deficient mice are very susceptible. This is measuring fungal loads in the mouth. So maybe Arid5a could be considered a potential target that would be selectively targeted perhaps for autoimmunity, and that its signals really tend more to an autoimmune pathology and not so much host defense. Again, very speculative, but I think it does illustrate the principle that not all signals are doing the same thing in all conditions. So finally, the last part of my talk, I'm going to tell a third vignette about a third RNA binding protein that really pulled us in some unexpected directions and opened up a whole new vista for me about RNA biology. And this is looking at another gene on this list called IGF2BP2, also known as IMP2. And again, just spoiler alert, IMP2 is another RNA binding protein, and it turns out it regulates both of the CEBPs, CBP beta and delta, by a very novel mechanism. At least it was novel to me. So I showed you in the very beginning when I talked about Matt Reddy's early work that we had discovered that IL-17 induces CEBP delta. And you can see that here. This is now qPCR. IL-17 strongly induces CEBP delta expression. But various people in the lab had attempted to understand how that was happening, and we really hadn't come up with a very good explanation. Rami Bashara was a postdoc in the lab who's now an independent assistant professor at the University of Paris, and he demonstrated that IL-17 enhanced the stability or the longevity of the CEBP beta RNA. And this is RNA decay assay in which you treat cells with actinomycin to look at the intrinsic decay. And the baseline half-life of CEBP delta is about 90 minutes. And this is increased approximately 50% in the treatment when you treat cells with IL-17. And this leads to a very rapid accumulation of protein. You can see in about 3 hours, we have a lot of CEBP delta protein that is increased in these cells. So how is this happening? And it was not obvious, even at the beginning, that the 3' UTR of CEBP delta didn't really have a lot of the hallmarks that one might expect in an intrinsically unstable RNA. But what Rachel Bailey and Rami showed was that there were sequences in the 3' UTR that corresponded to consensus sites for methylation. And I think it's not as well appreciated in immunology, perhaps as it is for DNA, that RNA can also be post-transcriptionally modified with methylation sites. And the most frequent of these is methylation at the N6 on adenosine at the nitrogen 6 called M6A. So there were, and so this process is mediated by enzymes that add these methyl groups called writers. This is usually contemporaneous with transcription, but it can be dynamic and it can be regulated by removal with demethylase enzymes known as erasers, the most common of which is FTO. And finally, this modification can be recognized by RNA binding proteins known as readers. And that's really what I'm going to talk to you about today. I won't show you the data, but we demonstrated that if we knock down either the writer or the eraser, that this influenced CEBP delta expression, suggesting that this pathway might be involved. And we also demonstrated directly that CEBP delta was in fact methylated at M6A. So what's the reader? And that really led us back to that array where we had IGF2BP2 on the list. As I said, I'm going to refer to it as IMP2. So what is IMP2? It was defined originally as a protein that binds to the 5' untranslated region of IGF2, and that's the origin of the name, controlling its translation. And it's been linked to regulation of thermogenesis. There are some GWAS links to type 2 diabetes. Many people have studied this in stem cell maintenance, cancer, tumorigenesis. But for us, the kind of aha moment came from this beautiful study that demonstrated that IMP2, in addition to these functions, is a reader of M6A. So M6A modification of RNA has mostly been studied with these canonical proteins called YTH domain proteins that tend to promote RNA degradation rather than to enhance it. But that the IGF2BP family, including IMP2, recognizes M6A typically in 3' untranslated regions and promotes stabilization. And so that really seemed to fit the bill of what we were looking at. These proteins can also influence translation, splicing, other effects as well. And this is a beautiful review if you want to know more. So when we knocked down IMP2, we found that this influenced a large subset of IL-17 target genes. Not all of them, but a lot of them. And including kind of our old friends, I hope by now, IL-6, CBP delta, lipocalin, a number of the CXC chemokines. I'll just point out CCL2 as well. It's going to come up in a minute. And so a large subset of IL-17 target genes not only had these consensus sites and were methylated, but were influenced directly by IMP2. Now, a number of things we did a lot of biochemistry. We struggled hard to appease the reviewers on this paper, and for which they made it a much better story. So I'm only going to show you one piece of data on this list, but we demonstrated directly that CBP delta mRNA is in fact methylated. IMP2 binds to this RNA at the 3' UTR, and that efficient binding by IMP2 does require methylation. So I'm just showing you that here. This is we used in vitro generated RNA probes that corresponded to the 3' UTR, either non-methylated or methylated, and looked at the ability of IMP2 to bind. And you can see at high concentrations of IMP2 that binding was fine. That's out here. But as we decreased the dose of IMP2, that binding really fell off. So it was much more efficient when we had the M6A modification than not, as you can see here. This methylation is required for stabilization of RNA. We map the sites. There are multiple sites. It's not quite as simple as one might think. And we also identified the binding partner who are as being involved in regulation of CBP as well. And so very simply schematic here that CBP delta mRNA is methylated. This is recognized by IMP2, and this is induced by metal-3, the methylase, and it can be downregulated by the demethylase FTO. So does any of this matter in vivo? And of course, we then looked at IMP2 knockout mice, which were very kindly given to us by Ning Dai and Joe Avrich at Harvard, and asked whether they were, these mice, for example, were they susceptible to AGN? And so looking, again, we measured blood urea nitrogen as a readout. IMP2, these are the littermate controls. Wild-type mice are subject to disease, and the IMP2 knockouts were substantially protected from disease. Not 100%. So ACT1 deficient mice, you can see, were completely protected, but they were very protected from the blood urea nitrogen and creatinine readouts, and really didn't show much pathology in kidney at all. And so if we look, for example, at lipocalin-2 expression in the kidney, that's very high in mice subjected to AGN and almost completely gone in the knockout mice. And the same thing was true for CBP delta. I'm showing you RNA here, but we showed this by protein as well. Many other parameters all in this paper. Not only that, but another IL-17 driven autoimmune model was also resistant, and that was EAE. So again, I think the audience is probably familiar with this model. This was done in collaboration with my colleague, Mandy McGeeky. And wild-type mice have a high clinical score, whereas IMP2 knockout mice were completely protected from disease. And we actually had this finding very early on, and it took us a long time to figure out the mechanism, because at first we thought, well, it's going to be IL-6 because we know IL-6 drives Th17 cells, and it wasn't. However, Th17 cells were not recruited to the kidneys, the draining lymph nodes, in the context of EAE. And this is shown here by flow cytometry. I will point out this is the only FACS plot I'm showing in this entire talk. So that's, to prove that I can have creds as a real immunologist. But what we came to was that, in fact, not only were Th17 cells influenced in the absence of IMP2, but monocyte recruitment was also impaired, and that turned out to be dependent on a lack of CCL2 in the stromal lymph nodes. I don't have time to go into this story in any more depth, but it just came out, and I encourage you to take a look at it. I'm sure the story is, in the end, more complicated than just CCL2, but I think it does illustrate that we have IMP2 affecting two different autoimmune conditions mechanistically in quite different ways. Finally, one might ask, well, could IMP2 be targetable? And up until just a couple of weeks ago, there were no reports of potential inhibitors to IMP2. And so we sort of took a different approach to seeing if blockade of IMP2 might have some sort of therapeutic potential. And to do that, we crossed IMP2 floxed mice to a constitutive tamoxifen-inducible CRE, the ROSA 26 CRE ERT2, and applied tamoxifen after inducing disease. And we did this for both AGN and EAE. For AGN, we started at day 1 after disease. For EAE, we started at day 7. And in both cases, blockade reduced disease pretty much down to what to very substantially reduced pathology and disease. So you can see that for the blood urea nitrogen for AGN, and you can see that in clinical score for EAE. And I think this nicely recapitulates what we would do in an inhibitor. It's not tissue specific, but when you give a person a drug, that's not tissue specific either. And so at least hypothetically, this could be a potential therapeutic. So in the end, these signaling pathways, this is still a very simplified picture. I didn't throw up the, warts and all, every possible pathway. But if you want that, you can take a look at this review that we published a number of years ago. This is a genuine journal cover. So, every now and then you succeed if you persist. But I think the take home story that I would like people to take home is, all of these things are a balance. You need enough IL-17 around to keep infections at bay. And those include things like Candida, but Klebsiella, Staphylococcus. But too much is pathogenic. And that can come from overproduction of IL-17, which can be stimulated in unfortunate cases by these commensals, as we saw in the case of AGN. But it can also come from a failure to restrain signaling. Too much signaling either by a lack of inhibitors or by overexpression or overactivity of positive activators. All of these things fine-tune expression, and there's a lot of crosstalk. So maintaining that knife edge it is challenging, and it sometimes it causes me to wonder how any of us are walking around alive today. So finally, I think I'm pretty much on time. I want to thank all of my wonderful lab members. This is the current lab, and if anyone's interested in joining, please send me an email or contact me on Twitter or whatever. Always looking for new faces. Many fantastic former lab members. I think I tried to call them out as we went along, and amazing collaborators. A big special call out to my 2 colleagues, Partha Biswas and Mandy McGeechy, who've really been a lot of fun and a pleasure to work with. Finally, I'll use this platform as a shameless plug for the International Cytokine and Interferon Society meeting, which is in Hawaii in September. This is the website, and we're all starved for meetings, so come join us in Hawaii in the fall. With that, I will say thank you.
Fantastic, Sarah. Can you just stop sharing, and then we'll put up? Yeah, I wanted to remind people. Yeah, so first of all, that was really beautiful. I love the integrated discussion of transcription and RNA stability and translation, all sort of feeding in to regulate these responses, and really intriguing that you can sort of target specifically perhaps the bad side of IL-17 without perhaps compromising antimicrobial defenses. So super elegant, really beautiful. Okay, so that was really fantastic. So as everybody knows, the way we do the questions here on the Global Immunotalks is through Twitter, and I know Sarah is an active tweeter, and her Twitter account is listed there at the bottom at SLG1717. So please look for the Global Immunotalk site, and there will be a tweet that says ask questions for Dr. Sarah Gaffen here, and then Sarah will respond, and hopefully there's some active discussion on there, and remember to sort of mention and tag the Global Immunotalk site. And I think that is it. So thank you very much. Just a reminder again for joining in next week for next week's speaker, and we'll see you sometime soon. Thank you, everybody. Thank you, Sarah.
Thank you.
Yeah, that was wonderful. Bye-bye.
Bye.